Okay, guys, let's talk about Lab 1. It's going to be about the light microscope. Now, microscopes, um, this is really something you need to be hands on. But since we can't do that, we've got some pretty good stuff at the end of this. Let's talk about in general. So, some things are just too small for us to see with our eyes. So, what we have to do is get magnifying lenses and use those to make the image appear larger. And that's basically what a microscope is. Now, we've got two lenses, one in the eyepiece and one in the objective lens. So we multiply those. The lens that we put our eye on is going to magnify 10 times. The lenses that are on the bottom on the objectives, they'll either magnify 4, 10, 40, or 100. So let's say the lowest one, 4. If I use a 4x objective and I use the 10x on the eyepiece, that's 10 times 4 is 40. That's kind of how what we got. You can see right here they're talking about that. We have the 4 objective lenses and the binocular or the eyepiece. We've got a movable stage. We'll talk more about this when we get to the picture, okay? Now, a light microscope will let us look at something that is as small as 2/10 of one micrometer across. A micrometer is 1/1,000,000th of a meter, super tiny. But they have to be, if they're any smaller than that, then it's going to kind of all run together for us. Something have to do with resolution. So we need things to be magnified. We need them to look bigger than they really are. But they also have to have resolving power, because if we can't distinguish between two things, then they all look the same. Think of it this way: if you've ever been on a road and saw in the distance maybe trees, if you're so far away you can't tell the individual trees, it looks like one big mass. But the closer you get, the more they're magnified. You can resolve those and say, oh, that's one tree and that's another. And it's kind of the same thing with this bacteria that we're going to be looking at. We already talked about this. If I want to know how much I'm actually magnifying, I take the eyepiece, the ocular lens, that's the same thing, 10, and then I multiply it by the objective lens. So 10 times 4 is 40, 10 times 10 is 100, 400, and then 10 times 100 is 1,000. Okay, let's see what else we got, guys. Resolution, we've already talked about how to tell the difference between two objects that are closely spaced together. Resolution, when you get to the 1000 magnification, in other words, when you use the 100x lens, that resolution, that magnification is so strong that you actually have to put a drop of oil between the objective lens and the slide, or else you won't be able to see anything. It'll be all, the light will scatter. The only way we can keep it straight and get a good image is to put a single drop of oil that's right there on the objective lens. So there's no actual air gap between the slide and the end of the lens. We use the oil to bridge that gap. Okay. Okay, I think we may have mentioned this. Maybe no, I'm sorry, it was A&P1. As far as carrying the microscopes, you always put one hand around this part right here, one hand under the base. That's how we carry these guys. Already talked about that. Always carry it upright. Now, there are several kinds of papers in the lab. You have lens papers. Guess what? Those are for lenses. Makes sense. You've got chem wipes. Those are not for lenses. Those are for wiping off slides. We only use lens paper. Any other kind might scratch or leave fibers behind. Um, and we also use, since the lens paper is absorbent, we use it to get the oil off the slide, off the bottom of the objective, the 100X. And by the way, just as an aside, the only lens you can use with oil is the 100X. It's sealed so that that oil won't get inside and permanently fog up those extraordinarily expensive ground lenses. So that means once you put oil on a slide, you do not go back down. You have to stay on the 100. And that's going to be important when I tell you exactly the procedure for getting a good picture or a good view through a microscope. We always put the cover on it when we're storing it. It keeps dust off of it, makes it easier. Yeah, we always number our microscopes. The cords on the back of the microscopes is actually a place right back here to store those. You wrap them up and put a little Velcro strap around it. The only thing we can do as far as those lenses is to remove the dust and the oil. Hopefully you won't see things like fingerprints, but you don't know who's had that microscope before you. So you will definitely need to clean it off. Again, guys, I realize you guys aren't in the lab this semester, but I've got to go through it just like you were because they're going to require you to know how to do this. Oil you can blot off, dust you just kind of wipe gently. Okay. And you turn off the light switch on the microscope before you unplug it. A lot of times the lamp will blow, but a lot of times it's simply something, a loose connection. So if you have trouble with that, you always talk to your instructor. Okay, so parts of the microscope. There's a link at the back of total end of this guide that will give you some sites you can go to, but I'm going to go through it too. Those are the eyepieces or the ocular lenses. Diopter adjustment, that's not something you're going to do a lot of. That's the arm of the microscope, and this guy right there, that little round piece, is the nose piece. Coming off the nose piece are those objective lenses. There's 4 of them generally. That's the stage, and there's some clips on there, and the stage is where you put the slide that you're going to be looking at. Underneath the stage, you'll see a condenser and an iris diaphragm. The condenser, for our purposes, we just move it all the way in one direction and leave it. The iris diaphragm you're going to have to use, you're going to have to adjust as you go from the 4X to the 10X and so forth. There's the lamp. It's just an electrically powered light that is going to shine up through here and come up through this iris diaphragm through the hole in the bottom of this and shine light on the slide from the back. There's the on-off switch. They're not showing the light switch on here. Most of these have a little slider, a little wheel that you turn, and as you turn it, the lights gets brighter or dimmer. This is the way you move the slide or the stage up and down. You've got a coarse adjustment knob and a fine focus knob. The coarse adjustment is going to move the stage a good significant bit at one little time. The fine focus is going to move it just very tiny, just fine adjustment, which hence the name. So guys, here's a rule of thumbs. You don't use the coarse focus except on the 4X and the 10X. When you get to the 40x, you should never use the coarse focus. You only use the fine focus. Because if you do, then you're going to go crack. You're going to break the slide. Not a good thing. The base of the instrument. Okay. I'm not going to read everything to you. I really don't think that's totally necessary, because I've told you most of what's going on here anyway. So if we were going to do this in the actual in the lab, we'll give you some slides, pre-made slides of bacteria, and this is an algae. And you could put those on there and practice using the microscope. And then you would write down in your results section what's going on. Now, here's how you use the microscope. First off, I would turn the light switch down somewhat before I first start, because on the 4X, that light is going to be very bright, coming up and hitting your eyes, and it'll hurt your eyes a little bit. So turn it down, put the slide on the stage, use the coarse adjustment knob to bring it up while you're looking through the lenses, right? Through the ocular lenses. And when you get it as good as you can, you stop there, and then you start using the fine focus to get it as sharp as possible. The next thing you do is make sure that that guy is totally in the center. What you want to look at needs to be in the exact center. It's going to look like a little circle of light. That's what you're going to see. Needs to be centered. Why is that? Because when you go up, that circle of light is going to get smaller, and if it's not centered, you're going to lose it. So once you've got it perfectly adjusted, centered, and you've got the sharpest picture possible, then you go on the nose piece and you rotate it from the 4X to the 10X, and you do the same thing. You can use the coarse adjustment. A lot of times you won't need it, but you'll use the fine focus to regain that sharp, clear image. You're going to make sure that you've got this guy totally centered in your little white circle of light. Now at this point, you can actually try changing the light on that little dimmer wheel, because you are going to have to change it as you go up. You're actually going to need more light as you go up, but you got to be careful. If you put too much light, it's just going to white everything else. It's going to be glare, and you're not going to see anything. Okay, and at this point, you can, although to be honest with you, on the 10X, you probably won't need to do much with the iris diaphragm. Okay, once we've got everything perfect, then we go up to the 40X, and we do the exact same thing. But this time we know that we don't use our coarse adjustment knob, only the fine focus. We use that fine focus, get the sharpest picture, and we're also going to have to use that light dimmer wheel. We're going to have to try moving it back and forth until we get the right amount. And at this point, you probably are going to have to reach around. That iris diaphragm depends. There's a way of adjusting it. Depends on the microscope. You want to try to move it and see what effect that has on your picture. Guys, I can't tell you an exact thing, because this is called tweaking. You just make teeny tiny fine adjustments until you got it perfect. Now, you're on 40X. You've got that guy centered again. You've got a perfect picture. You got to make sure it's perfect, because when you go up to the 100X, you're going to need oil. Once you put oil on that slide, you can't go back. Now, if everything is good, then you move the objective lenses. You move it between the 40 and the 100X, and you put one single drop of oil on the slide, on that circular area on the slide where the specimen is. Once you do that, then you rotate it the rest of the way and put the 100x over the top of it. Then you're going to need to adjust the fine focus. You're going to need to try the light dimmer, or actually more light, and you're going to need to try to adjust the iris diaphragm. This is a lot like learning to ride a bicycle. I can tell you you have to balance this way and that way, but truthfully, the only way you really can do this is to get on there and do it. Now, we do have some good, excuse me, we have some good simulations that are going to do much of the same thing. You're not actually going to have your fingers on a knob, but you're going to be required to go through these same steps. That's one of the reasons I'm telling you about it. Okay, I think this whole thing is basically what I have said. They're telling you to draw this. Of course you would if you were doing an actual notebook. Okay. Once you're finished, the thing you've got to do is to clean that oil off the slide. So this, in this case, remember slides, chem wipe. I don't know why I was wanting to say kiwi wipe. Maybe I'm hungry, who knows. But in any case, you use a chem wipe to wipe off the slide. You use a lens paper to wipe off the 100x, because it will have oil on it because the oil was touching the slide and the lens. You've got to blot it and make sure you get all of it up, because if it's allowed to dry on there, it will cloud that lens. And again, these things are way expensive, guys. When you get ready to store this guy, you guys may have noticed that the 4x is the shortest objective lens and the 100x is the longest, which is why you don't use the coarse adjustment knob. But when you get ready to store it, you want to move the nose piece until the 4x is the one that's down over the stage. Then you're going to take that stage and you're going to move it all the way down to the bottom. You unplug, turn the light down, of course. Turn the actual power switch off. Sometimes those are the same thing, depending on the microscope. Then you unplug it, rewind the cord, put it in there, put the cover back on it, and you replace it up. Well, guys, that's everything on Lab 1. This is really good. That's those amoeba sisters I told some of you guys about. This is really good. It's going to give you a way of practicing doing those steps. This is a really good as far as the parts of the microscope. A little practice. And this is another simulation. I would do both of these. Now, in order to get here, you can see when I hover my cursor over it, you do Control and then you do a left mouse click and it'll take you there in there. Okay. What I want you to do after you do this, I want you to answer these questions here. And I'm thinking this is what we're going to count as your lab notebook for those 10%. So answer them to the best of your ability. Okay? And that's got Lab 1. Lab 2, I'll give you guys, and that'll be our first week's labs. Thanks, guys.